Good afternoon. My name is Josh Huntsberger, and I'm Executive Director of Regenerative Medicine Manufacturing Society. I'm joined with my colleague, Ann Murphy, who's Executive Editor of Stem Cell Translational Medicine. And we both want to provide a warm welcome to everyone who has joined our webinar on gaps and opportunities for fighting COVID-19. We have a very exciting program for you all today. And if colleagues of yours have missed this webinar, we will be recording it and making it available on Stem Cell Translational Medicine's Stem Cell portal page. But we do encourage everyone to attend the entire webinar, as the last segment of this webinar will be a live panel discussion with many leaders in the field of regenerative medicine. There was an opportunity upon registering to share questions with our moderator. We've sent those questions, and we will try to address these questions and more during the panel discussion. We also have planned a perspective article that will follow this webinar that will summarize some of the key issues discussed as to what the field of regenerative medicine is doing to address this pandemic. Next slide, please. So our roadmap for today is to provide a little bit of background on Regenerative Medicine Manufacturing Society and how you can join some of our working groups. We'll also highlight our official journal, Stem Cell Translational Medicine, and the regenerative medicine ecosystem we're building through the Stem Cell portal page. This ecosystem is specifically focused on facilitating collaboration in regenerative medicine to accelerate research in fighting COVID-19. We'll also cover another important resource that Stem Cell Translational Medicine has developed, which is a clinical trials database, which lists clinical trials by condition, which can be further filtered based on type of intervention, example drug, or combination product. We'll also cover a vision for creating knowledge hubs to improve this clinical trial database, highlight some of the resources that have been submitted to our Stem Cell portal page, and then segue over to the keynote talks, which cover what leading institutions are doing in the field of regenerative medicine to fight COVID-19. Next slide, please. The Regenerative Medicine Manufacturing Society is the world's first and only professional organization dedicated solely to advancing the field of regenerative medicine through manufacturing. Its mission is to identify unmet needs and gaps in regenerative medicine manufacturing and catalyze the generation of new ideas and solutions by partnering with private and public stakeholders. Society members who represent industry, government, academia, nonprofit organizations share knowledge and work together to create solutions to manufacturing challenges. The ultimate goal is to develop standardized manufacturing processes to facilitate the smooth and quick transition of new therapies to market for the benefit of patients. The Society provides a platform through working groups that focus on 4 primary areas: cell and biomaterial manufacturing, standards, 3D bioprinting, and artificial intelligence-enabled automation. Showcased here is a perspective article that came out of one of our working groups, and it addresses how regenerative medicine is uniquely positioned to fight COVID-19. And one of the resources that's highlighted in this article was developed by Stem Cell Translational Medicines and is highlighted on their portal page, which we'll hear more. I'd like to turn over to my colleague, Ann, to talk a little bit more about the journal.
Thank you, Josh. I also am delighted to welcome all of you to this webinar. Thank you for joining us. So this year, 2021, is our 10th anniversary of Stem Cells Translational Medicine. And we're very grateful to CIRM and also to Quintiles, now IQVIA, for their founding grants that made the launch of this journal possible in 2011. So for those of you not familiar, who may not be familiar with the journal, our mission is to bridge stem cell molecular and biological research and to hasten the translation of emerging lab discoveries into clinical trials. To move applications of these critical investigations closer to accepted best patient practices that ultimately will improve outcomes, and to advance the utilization of cells for clinical therapy. So the journal focuses are these. Stem cells therapies and regenerative medicine have the potential to eventually repair or replace tissues with damage from disease. Clinical translation requires many areas of expertise. So from preclinical research to technology transfer, process development, regulatory adherence, manufacturing, quality assurance, quality control, quality, trial design, and human trials. During our first decade, most of the cited articles are mainly representative of the evolving process we've seen develop through research worldwide, and they cover three major areas: stem cell research, stem cell byproducts, such as extracellular vesicles, and research that is advanced to human clinical trials. We're very pleased to say that one index of our maturation over this decade as a globally recognized peer-reviewed journal is that our impact factor is 6.429. So in addition to the monthly publications, the journal is the official journal of our MMS, and we've established a regenerative medicine community to facilitate global and vibrant network of academic, government, including regulatory and industry members, all working together to advance the field. Next slide. The stem cells portal, I'm sorry, Josh, Nick, if you go back, the mission of the stem cells portal is to build and maintain a stem cells and regenerative medicine network. We're developing the portal as a shared platform to provide coverage of the latest research and news, from bench science to developments in clinical applications. So in response to the global pandemic, the journal and RMMS have partnered to build a freely accessible collection of shared resources across three areas: models, cell therapies, and technologies, to help bring the scientific community together and to highlight how the field of regenerative medicine is uniquely positioned to fight the COVID-19 pandemic. The resources listed here are developed and submitted by scientists working on regenerative medicine long-term solutions for this pandemic and the next. And we encourage, as Josh has already mentioned, we encourage everyone to submit their resources. We envision highlighting these resources through the portal, through webinars such as the one today, through publications that Josh has mentioned, and other scientific meetings. The importance of clinical trials, next slide, please, and reporting of data, reporting the data of trials, regardless of whether they've met their endpoints, cannot be overstated. In addition to our encouraging clinical investigators to report and publish their data in our human clinical articles journal section, we've created on the stem cell portal a section called Clinical Trials, Stem Cells, and Regenerative Medicine. And next slide, please. And as Josh has mentioned, this Stem Cells and Regenerative Medicine clinical trials helps investigators navigate through over 5,000 registered trials involving stem cell research on clinicaltrials.gov. By developing a clinical, what we've done is develop a clinical trial listing, which is actually an RSS feed based on presenting clinical condition categories. Clinical trials are organized by condition with the ability to filter results by intervention type under each condition. And further information, of course, can be found directly on clinicaltrials.gov. And Josh will talk about this clinical trials listing as one of the initial steps in the slide that you see on the screen with our MMS, the journal, and Oracle Knowledge Hub vision. Thank you.
Thanks, Anne. Yes, and so just, Matt, can you back up the slide to stay on the hub? Great. And so in a collaborative effort with RIMS, Stem Cell Translational Medicine, and Oracle, we have a vision to create a data access platform across the development lifecycle of regenerative medicine product that links together data with scientists, clinicians, regulators, companies, investors, and funding agencies to ensure a first of its kind knowledge hub that will really accelerate the development of regenerative medicine products and reduce redundancies and duplications of efforts. Next slide, please. And so before we start our main program, I did want to highlight some of the regenerative medicine resources that are already up on our stem cell portal page. And this can be found stemcellportals.com. And they're categorized in 3 areas. So for models, here's just an example. Wake Forest Institute for Regenerative Medicine has developed a body-on-a-chip platform that has demonstrated exceptional performance in modeling normal and pathological human physiology. In cell therapies, there's an approved clinical trial about to begin recruitment for the management and treatment of post-COVID-19 lung damage. And for advanced technologies, again, here's just an example. One company, Biospherics, has a next generation CGMP compliant isolator that can provide physiologically relevant gas levels and temperature for cell incubation, as well as cell handling, and is already in use for generating human cells for COVID-19 clinical trials. So I do urge you to please visit this site, stemcellportals.com, and upload your resources if you do have ones that fit these 3 areas: models, cell therapies, or advanced technologies. Next slide, please. And so with this, we have a very exciting program today where leaders in the field, which you can see here, will be providing updates on how regenerative medicine can be used to battle this pandemic. And this will be a pre-recorded video that'll last about 40 minutes, but it will go through each of these highlights. Matt, can you start the video, please?
Hello, it's great to be with you today as part of this workshop where we will discuss some of the tools that we have to fight COVID-19. Stem cells translational medicine had a leading role in disseminating information about COVID-19, and in August, this paper was published about how to fight COVID-19 using regenerative medicine therapeutics. You know, regenerative medicine really is poised to provide a solid understanding of infection mechanisms, not just for this pandemic, but for future pandemics to come as well. And to be able to screen agents for toxicity, testing, and efficacy, and to introduce innovative treatments using these strategies. When we look at COVID-2 specifically, we have to realize that it's attacking many different organs where regenerative medicine really does play a role, including the lung, liver, heart, kidney, and there's 4 times a higher chance of stroke. So what are the tools that we have available? First of all, we have our in vitro model organoid systems where we use regenerative medicine techniques that can actually be better than the current standard, which are 2D culture systems and animal models that do not necessarily reflect the actual human response. By creating what's called the body in a chip, we can then start to create miniature organs, just like we would with the same strategies that we use for using organs and tissues in patients, where we take the same type of normal primary human cells in the same proportion, and we mix them with the glue for each specific organ to create very sophisticated structures that, for example, in the liver, these miniature organs even have a primitive biliary system. For the hearts, they can also be made with normal human cells, and they beat at 60 beats per minute. These can be kept alive for 6 months. And going through this process, you can give drugs that give you, drugs that actually provide a speeding up of the drug or slowing down of the heart using different agents at a drug dose relevant response. You can even create miniature heart attacks that can replicate what the COVID virus does. Miniature brains have also been created with all the cell types that the brain has, all the major 6 cell brain types with a normal intact blood-brain barrier that can mimic stroke, another condition that COVID attacks. The lung, of course, is the most important. And again, the 3D organoids are able to replicate the 3-dimensional structure that is not possible with 2D cultures. And it mimics the environment that is present with acute respiratory distress syndrome, which is a major component of COVID activity. You can see that these can be infected with bacteria and viruses, and it has also been infected with the COVID virus, where we have been able to show infectivity of these lung structures and how this is better than 2D culture systems. And we've also been able to use the system to test antidotes for drug development and drug discovery for COVID-19 and other future viruses for pandemics. Another part of our tools for regenerative medicine includes 3D printing. So for example, 3D printing can be used to print these miniature chip structures. In this particular case, we're printing the whole structure together, the chip and the organoids together. This is very small, about the size of 3 dimes stacked together. In this particular chip, we have liver, kidney, and ovary. We can use testes to make it male, female, or both. We can use 6 organs together, like in this chip, where we have liver, heart, intestine, lung, blood vessels, and testes. You can even do a module with 12 organs together on these microchips. Why are this important? Because this system is very sensitive. Let me give you one example. This is a drug called Hismanol. It was in the market for 11 years before they were able to connect the dots that this drug that had been approved by the FDA and had shown safety in cell lines, animal models, phase 1, 2, and 3 clinical trials turned out not to be safe. Took 11 years to find the common thread that led to showing that this drug was actually causing heart block. The drug was pulled from the market. We obtained the drug. Within 2 weeks in this microchip system, we were able to show the drug was in fact toxic to the heart, same mechanism of action, same human relevant drug response, because these systems are very, very sensitive, and we can do the same thing for drugs that fight COVID. In fact, we have now launched what we call the Humanoid Sensor Consortium, which is to develop artificial intelligence machine learning capabilities to accelerate drug screening and discovery. By cataloging the data from every step of the process, one can use artificial intelligence to identify patterns across these large, robust, integrated data sets to give us predictive modeling for future drug development against COVID. This consortium has membership from industry, government, and academia, and our funding member is Oracle, who's contributed Steep Cloud and will help to manage aspects related to AI, and we've already started to use this system for COVID-19. Another tool in our toolkit are stem cells. As you know, mesenchymal stem cells are the leading potential cell therapy candidate for acute respiratory distress syndrome and COVID-19. These cells are able to modulate overactive immune and hyperinflammatory processes. They also promote tissue repair, and they are also anti-infective. So these cells have been used in many different manners, but also byproducts of these cells can also be used for regenerative medicine therapies. There's a whole armamentarium of different types of cells and cell byproducts that could be used for COVID-19, and a lot of these are currently in experimentation and in clinical trials. Specifically for stem cells, their immunomodulatory effects are very powerful. They have been able to secrete endosomes, and they can reduce damaging inflammation by reducing inflammatory cells and cytokines. And they can also help to fight infection and give the lung tissue a resistance to lung infection from COVID-19. And there are more than 300 related clinical trials using mesenchymal stem cells for COVID-19 currently that have been registered in clinicaltrials.gov. In fact, one of the papers was published by our journal, Stem Cells Translational Medicine. This was just recently published, umbilical cord mesenchymal stem cells for COVID-19, where ARDS was treated in a double-blind phase trial showing improvement of this treatment for patients with COVID-19, showing the power of these stem cells. So to summarize then, we have a lot of tools in our toolkit to fight COVID-19 using regenerative medicine strategies. We discussed in vitro models such as organoid systems, we discussed 3D bioprinting, and we discussed stem cells. There are many other tools, but these are the major tools that regenerative medicine is currently using. So I'd like to thank you for your attention today and to remind you to please join us for the 2021 joint World Stem Cell Summit, Regenerative Medicine Foundation, and Regenerative Medicine Essentials course at Wake Forest on June 14th through the 17th. And of course, this event, which is an annual international event, is sponsored by our host, our journal, Stem Cells Translational Medicine. Thank you for your attention.
Good afternoon. My name is Martha Lundberg, and I'm a program director at the National Heart, Lung, and Blood Institute at the NIH. I would like to thank the Regenerative Medicine and Manufacturing Society, and especially Dr. Hunsberger for inviting me to this important and timely webinar. I would also like to thank my colleagues for their input on this presentation today. It is important to highlight several items with regards to NHLBI efforts for fighting COVID-19. These include NHLBI administrative and revision supplements, accelerating COVID-19 therapeutic interventions and vaccines, or ACTIV, rapid acceleration of diagnostics, or RADx, and also I would like to note about the Office of Research Infrastructure Programs, or ORA. With regards to the NHLBI administrative and revision supplements, there was a very large, robust response. 11 awards were given for a total of $6.2 million in fiscal year 2020. And the awards covered all areas of the heart, lung, blood systems research. Some of the overarching goals for NHLBI responses to the pandemic are preclinical, and this is in regards to strategic objectives to accelerate testing of compounds. These include a preclinical resource map that would provide a master inventory of testing resources, a pandemic response network framework in order to create a national strategy for resources and establishing a virtual preclinical in vitro and in vivo testing network. Prioritization is important because this is about establishing standard procedures and data to support preclinical work. And finally, NIH would like to create a public database for sharing of in vitro and in vivo model characteristics and data. This slide shows the active clinical working group activities. There are 5 active programs. Active 1 is testing promising immune modulator compounds, class drugs, that help minimize the deleterious effects of an overactive immune response to SARS-CoV-2. This phase 3 trial will enroll hospitalized adults with moderate to severe COVID-19 disease. Active 2 is a master protocol designed as a phase 2 trial that can expand to phase 3. NHLBI is not involved with this clinical trial, but you can find additional information by clicking on the above referenced website. Active 3 is designed as a phase 3 trial with 2 stages of testing, allowing for quick analysis of compounds for effectiveness, and then a progression to stage 2 clinical trials to validate the compound for broader use in patients. Active 4 is an outpatient protocol and investigates whether anticoagulants or antithrombotic therapy can reduce life-threatening cardiovascular or pulmonary complications in newly diagnosed COVID-19 patients who do not require hospitalization. Finally, somewhat new, the Active 5 program is designed to conduct a series of randomized, double-blind, placebo-controlled phase 2 trials using common assessments and endpoints. Trials using this protocol will enroll hospitalized adults with COVID-19 to evaluate whether certain approved therapies or investigational drugs in late-stage clinical development show promise against COVID-19. Here, I would like to discuss the overall NIH coordination of the NIH RADx program for the COVID pandemic, in which NHLBI is participating in multiple levels. I would like to thank Dr. Machado at UCSD for the following slide contribution. For the RADx Data Coordinating Center, Dr. Spencer and Machado at UC San Diego are the major PIs, as well as Dr. Zhu at the University of Texas. They provide important expertise and resources to the RADx program and overall coordination. Here's the structure of DC3PO, or the Data Coordinating Center, which consists of a central data discovery component and the coordinating center. The program will be governed by an external advisory board, training committee, and steering committee. The steering committee will receive direct feedback from the Data Safety and Monitoring Board for any specific safety concerns. RADx-RAD awardees will be able to interact with Dr. Spencer and Dewey with regards to data use and diagnostics and data collection, integration, and sharing. Finally, this work will flow down to the use of FDA-approved tests, standard samples, as well as data indexing, privacy, and security and integration. This slide shows the overall geographical distribution of the RADx initiative at the NIH. We announced 13 funding opportunity announcements and have 49 awardees, including the Data Coordinating Center. NHLBI is engaged with the overall coordination of the RADx program through DC3PO. We are also supportive of several targeted initiatives as listed below. These include the automatic biosensor detection and tracing program, as well as novel biosensing programs, and some others which are important, including the Prevail Kids program and the Exosome-based Technologies program. To wrap up, I would like to touch base on the coordination of the NIH COVID testing studies using our non-human primate model resources. Importantly, folks should be aware that the use of the National Primate Center resources must go through the ORIC portal for approval. This includes studies coming in under the active program as well as other COVID-related programs. Many thanks for your attention, and my contact details are here. Thank you.
Good afternoon, everyone. I'm delighted to be here with you this afternoon. Thank you very much for the invitation. My name is Lucie Lowe, and I am a Scientific Program Manager for the National Center for Advancing Translational Sciences, or NCATS. I'll be talking to you today about how tissue chips are being used to address COVID-19 challenges. So first of all, what are tissue chips? So tissue chips are bioengineered devices that mimic the key functional aspects of organs and tissues, human tissues. And a couple of examples here on the screen. On the left-hand side, we see a tissue chip in the palm of a glove that was on the front of National Geographic magazine a couple of years ago. And on the right-hand side, another example of a tissue chip with the coin for scale, so about the size of your smartphone. So showing the differences in size here. But they can be used because they're helpful to model diseases and test drugs and therapeutics in vitro, so before they get to clinical trial stages. Why are they helpful? Well, development of new drugs nowadays is very slow and costly, and so we desperately need alternative tools to improve the therapeutic development progress, and tissue chips can be very helpful along that scale. By the way, tissue chips are also known as microphysiological systems, or are synonymous with organs on chips. Tissue chips are also able to be used to respond to global health emergencies, though, and that's what I'll be telling you about today. So last year, NCATS was able to rapidly issue funding to our researchers through the US CARES Act to develop tissue chip models for COVID-19 and to model viral infection, for example, to understand multiple tissues and organ pathologies in linked systems, to test candidate drugs and vaccines within these systems, and to try and understand the immune responses on tissue chips, as well as model complications from vulnerable and at-risk patient groups. So I'll walk you through some of the projects that we were able to fund through this initiative and give you some examples. So this slide is showing how we were able to fund projects that were looking at the molecular mechanisms of neurovascular infection, SARS-CoV-2 infection, looking at how there is infection travels from the lung to the brain, so between the lung and the CNS, obviously looking at modeling SARS-CoV-2 infection in the lung and the heart, looking at the effects of SARS-CoV-2 receptors, not just in the lung, in the primary sites of infection, but also in other tissues such as the kidney or the liver, looking at various patient subgroups, so for example, diabetic patients using liver tissue chips, looking at the cellular and molecular pathologies in various different tissues, such as the kidney, the heart, and various vascular tissues, and also even looking at SARS-CoV-2 infection in the gut in both humans and bats. So a couple of examples of the research that's being done during this time. Here's one example of a lung on a chip that is being rapidly repurposed to test therapeutics during viral pandemics. So this is a team that was already funded by the NIH to model human lungs on chips and look at the response to the influenza virus. So they were ideally positioned to start looking at SARS-CoV-2 infection instead. The team did this by introducing the virus to the air channel within their lung chip on the top right-hand side. You can see the schematic here. And you'll see that the lung epithelial and endothelial cells are separated by a thin membrane with an air channel above and a blood channel below. An introduction of a pseudovirus into this system was able to look at the viral entry through the membrane into the blood channel of these chips. So testing a number of existing FDA-approved drugs, the team were able to show that some drugs did seem to show promise for inhibiting viral entry, highlighted here in green. So for example, amidiaquin, and other drugs such as hydroxychloroquine did not seem to inhibit viral entry, which despite hype from last year was consistent with clinical data that was produced. So this project was really showing proof of concept that tissue chips can help identify existing drugs for repurposing during the SARS-CoV-2 pandemic. Although I will point out that this is currently published as a preprint and is currently undergoing publication in a peer-reviewed journal. Another project was looking at the effects of SARS-CoV-2 in other systems, in this case in the kidney. So our researchers at Harvard looking at kidney on a chip had realized that acute kidney injury can accompany SARS-CoV-2 infection. And so by looking at human kidney organoids infected with SARS-CoV-2 virus, they were able to show that through kidney injury molecule 1, or KIM1, which is a receptor in the kidney, this actually was facilitating entry of SARS-CoV-2 into the cells. And this middle panel shows some human kidney organoids that have become infected with SARS-CoV-2 virusomes, these granular objects within the cells. They were also able to show that when these cells were exposed to the SARS-CoV-2 spike protein, the kidney cells showed an inflammatory response, as shown by red here in the top panel on the far right. So what this was showing is that the KIM1 receptor in the kidney is a potential SARS-CoV-2 receptor that could therefore also be a potential therapeutic target for preventing or treating COVID-19. So here the team were able to show a proof of concept that tissue chips can identify alternative routes of SARS-CoV-2 infection and also give insights into the systemic effects of viral infection. Again, this is currently a preprint. Finally, I'd like to highlight some of the efforts that the NIH and partners are doing to help advance COVID-19 research. In this case, it's developing a working group called the MPS Core, which stands for the Microphysiological Systems COVID-19 Research Working Group. And this is a joint working group that is supporting and helping coordinate global MPS efforts to study COVID-19 and try and position them also for understanding future infectious diseases and applications in that area. So this is a partnership between a number of institutes at the National Institutes of Health, the US Army's Combat Capabilities Development Command Chemical and Biological Centre, or DEVCOM CBC, and the UK's National Centre for the Reduction, Replacement and Refinement of Animals in Research, or the NC3Rs, excuse me. So this is currently a group of about 60 members, including representatives from the pharmaceutical industry, developers of tissue chips, academics, and also representatives from various regulatory agencies. And we held our first webinar recently. The core objectives of this group are to try and raise awareness of the utility of MPS for COVID-19 research, and to facilitate connections between MPS developers and end users, to engage global regulatory authorities, and also characterize model performance and readiness criteria so that MPS may be deployed rapidly in appropriate situations. We also support the assessment of MPS against in vivo or clinical data to quantify and validate these tissue chip systems in this context. And finally, ensure the 3 Rs, or the replacement, refinement, and reduction of animals opportunities, are recognized. And I encourage you to check out more details at the hyperlink below. But with that, I'll end my talk today, and thank you very much for your attention.
Hello, everyone. My name is Mahendra Rao, and it's a pleasure to be here. What I'm going to be trying to talk to you about today in the next 8 minutes or so is about novel cell-based COVID-19 tools that we can make available through our partnership with various companies. The 4 companies that have made these things available are listed here in the slide. One of them is RxCell, which is a small startup based in California. Implant Therapeutics, which is another small startup. It's a subsidiary of Pencela and is based in Maryland, United States. Virna is a new startup based out of Toronto, and it's focused on making specific antibodies, in this case, specifically monoclonals and polyclonals to COVID. Pencela was the parent company for which I am the CEO, and this company has been involved in using iPSC-based technology and novel gene editing tools to allow us to be able to develop important tools using iPSC. As you probably have already heard, many different companies have been using either adult cells or iPSC-based cells for COVID-related research. Some important things that they have talked about, which I will not be mentioning, are the use of MSC for ARDS and other sort of inflammatory disorders that are associated with COVID. Nor am I going to talk about how people have used iPSC-derived lung epithelial cells as a model for looking at COVID infection. Those are reagents, and I'm sure other groups will talk to you about that today. What I'm going to focus on is the use of cells to deliver either active or passive immunization and what we have done with this. I want to emphasize right in the beginning, before I show you any of the other slides, as to this is not just us alone. And there have actually been a couple of papers from different companies and groups in China which have already tested this in humans. So this is not completely novel, but certainly it is an opportunity and a useful way to be able to use cell-based tools. As you know, there are many, many COVID-19 vaccine development programs in place. This is a small list as of May 18th that I abstracted from one of the papers that was published on this. And they've all highlighted the various issues that are there with vaccine development. There's the issue of delivery, the persistence of the antigen, how many antigens can you deliver to a particular cell, whether the response will be good enough because you don't have the antigen persisting for long enough for the response to be good in all individuals. There are issues with the kind of antibodies that are developed and antibody-dependent enhancement, and the absence of memory T cells and the possibility that COV-2 may use alternate mechanisms to enter T cells, which have low levels of ACE2, and in that case, might be like HIV, that you may not be able to ever develop any good antibodies. And as you know, the CD47 is one of the new antigens that has been described. So why think about alternative ways of being able to present antigen? And the reason why I wanted to show you this, or why we thought about this, was that it's hard to get DNA or RNA, which is how Moderna and Pfizer and others have been trying to do this, into cells. And it's hard to do this in vivo. However, if you could do this ex vivo and use a cell which has already been used for therapy, it should be quite possible to be able to deliver an antigen in reasonably large amounts and get the appropriate quality control that's required to be able to deliver an antigen reliably and consistently. And we know how to manufacture cells in bulk, and we know how to distribute cells worldwide in bulk, and the ability to harvest MSC is available worldwide. Here's a list of some things that have been done and a lot of data that's available on how MSC can promote adaptive immunity. And I won't walk you through the entire slide, just point out that there are multiple mechanisms by which it may enhance the immune platform as well. So you can imagine that the process would be relatively straightforward. Take MSC, grow them in culture, depending on how you harvested them, use standard gene delivery techniques, whether it's electroporation, virus, piggyback transposase, or adenovirus or episomal vectors. And then once you've delivered it to the cells, select the right population, grow it up, and then you have a cell which can deliver this. And of course, as long as you can code that antigen, whether it's an antibody or it's a viral protein for making a vaccine, both of these should be equally possible. So why, if this is possible with any cell type, why use MSC? And in particular, why use iPSC-derived MSC? I already told you one reason for using MSC is because they're widely available and they have the same characteristics that have made them useful for a wide variety of other indications as well. iPSC-derived MSC have the same characteristics as adult or perinatal-derived MSC, but they have two additional advantages. One is you have the ability to gene edit. And secondly, even though MSC are hypoimmunogenic, you can make them even more hypoimmunogenic or truly hypoimmunogenic in a scalable way. And I think this COVID-19 pandemic has offered an unprecedented opportunity for us to consider how we can take MSC-based cell therapy to the next level. So this was our solution to make the cells even more hypoimmunogenic. And this was based on work that had been done by Universal Cell and other companies, which had shown that you can really knock out class 1 and class 2 antigen. And while this is quite good in allowing the cells to evade the immune system, the active immune system, it really enhances the passive or innate immune pathways as well, which in this particular case is a good thing, because what it does is it activates the pathway so that you will develop a resistance or an antigen, and your innate immune cells and the dendritic cells will be allowed to present antigen. So the way we've done this is we've taken iPSC, engineered them to knock out class 1 and class 2, and then been able to take those cells and differentiate them into MSC. I won't go through the rest of the slide. You'll probably have a copy and you'll be able to go through it in detail in the interest of time. So now you can imagine it's a very straightforward way. You have MSC, they're hypoimmunogenic, you've knocked that out, and now you can overexpress any protein or antigen or an antibody that you may need to do that. So are there antibodies available to COVID-19? And the answer is yes. There are several monoclonals and variants of how people make monoclonals that are available. And again, this is a very busy slide. All I want to show you is that there are many companies that have moved this forward. And as you know, Regeneron and Eli Lilly have emergency use authorization for the antibody cocktails that they have been using. And those have been shown to be very useful worldwide. And the Regeneron cocktail has even been useful for the new variants of COVID-19 that have been identified, whether it's been the South African or the Brazilian strain or the new US 20C strain that has been identified in California. We partnered with a company called WFIRM, which is a spin-off from the University of Toronto's antibody screening facility, and they were able to develop several antibodies as single-chain antibodies as well as dibodies and various variants that are known, and we've been able to show that those can be expressed in MSC. And once they are expressed, that they can be delivered in a reasonable concentration in an animal model for this disease. And again, I want to emphasize, we're not the only ones to have done this before. We've actually borrowed from a company called TRT, which is also based in Toronto, which had shown that this was possible when they were looking at equine encephalitis. So the one next step I wanted to emphasize here, because there are several other companies which are working here, and some of them are members of the Regenerative Medicine Society, and that is that there are small antibodies which are made in certain species of camelids. These include llamas, and these are called nano antibodies, in that the heavy chain and light chain is in a single molecule. So it makes it much easier to make this. And of course, other people had this same idea, and they were able to show that you can get good neutralizing single chain antibodies made in llamas. And that means that we have one more simple way of being able to take antibodies, overexpress them in MSC, and be able to deliver them in high enough concentration to be able to be effective. Okay. So this is just the background. And I just want to summarize here to say, it was not here to tell you all that we've done with this, but to tell you what is available. And in this case, what's available are the iPSC lines, a protocol, or just available iPSC-derived MSc. And again, I want to emphasize not just from us, but there are several other companies which are working on this as well. Notably, a company called Cynata also has iPSC-derived MSc in bulk quantities. We have class 1 and class 2 null iPSC. I don't know of all the other companies which might have them. We have class 1 and 2 null iPSC-derived MSc. We have antibodies which have been made by Werner. And of course, as you know, you could get antibodies from several different groups, including Sorrento Bio or Vir, which is another small antibody generation company. And if you needed those, you could talk directly to Dr. Naji, or you could talk to the company Vedna. We've also looked at newly synthesized antibodies in llama, which have been available from several groups, including Creative Bio. As you can imagine, if you can overexpress, you can take a ACE2 receptor expressing line, or you can make lung epithelial cells derived from iPSC. And we have a partner based in India. It's a company called iStem, and there are several other companies, of course, who have made such lines in immortalized cells as well. And we have iPSC-derived MSc, which express spike and other COVID-19 antigens so that you can develop an in vitro assay for infectivity and protection in a dish, which would allow you to do screening in a very rapid way. So I'll end here and make sure that if you have any questions, I can answer them at a later stage. I want to thank you for the time and your attention.
Hello, my name is Suhail Talib. I'm a Director of Therapeutics at CIRM, and I'd like to provide a brief overview of the CIRM's COVID-19 program. So by way of introduction, CIRM is a stem cell agency which was created by the voters of California in 2004, which allocated $3 billion to fund stem cell research and regenerative medicine. Subsequently, in 2020, additional $5.5 billion were approved by the voters of California to continue CIRM's mission. That is, to accelerate the stem cell treatments to patients with unmet medical needs. Since its inception, CIRM has funded over 1,000 projects and 68 clinical trials, which has enrolled more than 1,200 patients in phase 1 to phase 3 clinical trials for a number of disease indications and utilizing diverse therapeutic modalities, including small molecules, biologics, and gene therapy. Now, CIRM's COVID program was approved by the CIRM board in March of 2020, which allocated $5 million for these programs. Now, recognizing the urgency to develop these treatments for the epidemic, these projects have shorter award duration and short-term deliverables. In order to launch these programs rapidly, weekly grants review and monthly board approvals were carried out, and in a short period of time, 20 programs were launched last year. Now, recognizing the urgency and rapid progress towards a goal is needed. In order to have rapid progress, readiness to the start the project is essential. So for example, our CLIN2 programs, which is a clinical stage program, the applicant must have an IND filed and approved, and 6-month goal is to complete the enrollment and data collection. Similarly, for our IND enabling programs, which we call as a CLIN1, the applicant must have cleared the pre-IND, and the goal is to file an IND within 6 months. Similarly, our translational and discovery stage programs also have very clearly defined entry criteria and the goal deliverables in 6 months. Another important requirement for this program is addressing the need of the underserved. As you know, COVID-19 has a disproportionate impact on minority and underserved communities. Therefore, for clinical trial stage program, plan for outreach and inclusion of underserved community was considered as an important review criteria. Similarly, the project plans were required to include advantages or challenges of the proposed study in developing a protocol or a tool that will serve all populations. Now, let me give you a few examples of the COVID-19 projects funded by CIRM. There are 3 clinical trials. Most advanced is a randomized multicenter phase 2 clinical trial to test the use of off-the-shelf MSCs for the treatment of ARDS, that's acute respiratory distress syndrome in patients with COVID-19. PI for this study is Mike Matei at UCSF, and since it is a randomized study, both COVID positive and COVID-19 negative patients are enrolled in this clinical trial. Second clinical trial is a phase 1, is the evaluation and characterization of the antibodies in convalescent plasma that is used for therapeutic use. This study is particularly important to identify the set of antibodies which are responsible for the therapeutic effect, so as to connect the dots, that which particular antibodies indeed are responsible for therapeutic effect. Now, this study is being carried out at CTFO by Dr. John Zaya. Our third study is a phase 1/2 clinical trial that CIRM is funding, is carried out by Cellularity. And this is an immunotherapy approach in which placental HSC-derived natural killer cells are used for the treatment of COVID-19. The scientific rationale for this approach is based on the fact that natural killer cells are the first line of defense against viral infection. Idea here is to boost the body's natural innate immune response against the virus. Now, our discovery and translational projects are utilizing diverse approaches. Shown on this slide are examples of cellular vaccine to augment the cellular immune response against the virus, which includes identifying HLA-restricted peptides to generate highly specific cytotoxic T cells against CoV-2. Another approach is antigen-loaded nanoparticles are used as vaccine. And third one is the use of stem cell to small molecules to stimulate the stem cells and engineered dendritic cells to generate cellular immune response. Other discovery stage projects are addressing disease pathology caused by COVID-19. Approaches employed include iPSC-derived tissue organoids to screen or test antivirals. This is an approach which Dr. Gampers at UCLA is taking. Second example is an application of a small molecule to augment regenerative potential of the stem cells to repair organ damage caused by excessive use of ventilators in patients with COVID-19. This project is carried out by Dr. Helen Blau at Stanford University. And lastly, Dr. Karen Chrisman at UC San Diego is investigating the use of pro-healing biomaterial for tissue repair in patients with COVID-19. In summary, 20 awards were launched since April of 2020, which includes 3 clinical trials and 17 discovery and translational projects. I hope this gives you some idea of the CIRM's COVID programs, and thank you for your attention, and I'll be happy to address any questions during the panel discussion period.
Well, those were quite impressive talks, and I do want to thank all of the speakers for taking time to prepare those talks, and really excited for the panel discussion, which will follow. Before we jump into that, I did want to highlight some of the work that New York Stem Cell Foundation is doing. And we have the pleasure of hearing from Dr. Rick Monsma, who's Senior VP of Scientific Operations at New York Stem Cell Foundation during the panel discussion. So he can elaborate on some of this work. So New York Stem Cell Foundation is doing innovative disease research in such areas as diabetes, cancer, neurodegeneration, and psychiatric. They're also developing pioneering stem cell technologies, which we'll hear about in a later slide. Next slide, please. So here is shown some of the efforts in creating patient-specific stem cells from blood or skin samples that can be used to model disease. This approach is being used in over 300 conditions, including major ethnicity groups are included in this. And patients can also be linked to longitudinal clinical data. So this is their biobank effort at New York Stem Cell Foundation. Next slide, please. So here's shown some specific efforts on COVID-19. So New York Stem Cell Foundation is making lung cells and other cells affected by novel coronavirus as a way to test existing and new drugs to help stop or prevent infection. And then next slide, please. And this last one, again, just highlights some more efforts. So what I wanted to draw attention to is Dr. Chen, who's using organoids to study the human cells that are susceptible to SARS-CoV-2 infection, and has already identified drugs that block the virus's entry into cells. There's also a rapid testing work that's being done that could be mitigate costs, such as $6 per test. And then there's also work by Dr. Rossi, who helped contribute to the SARS-CoV-2 vaccine created by Moderna Therapeutics. So with that, I'd really like to transition to our panel discussion, where we'll call all of our speakers into that panel discussion. And we will have Dr. Anthony Atala from Wake Forest Institute for Regenerative Medicine. He will be moderating this panel discussion. So it'll take a few seconds to get all of our panelists on, so bear with us as we make that transition to the panel discussion.
Well, it's so good to see all of you. What a pleasure to have you all on the panel today. And you know, what a nice discussion. I tell you what, first of all, it's nice to be able to see each other after not seeing each other in their other venues. So it's nice to see each other in Zoom. And also have this group of participants here with us today looking at COVID-19. The COVID era, who would have ever thought? Just you know, a year ago at this time, we didn't know what was going to happen. You know, 2020 became this year full of COVID and challenges, and it's great to see all of you presenting your efforts in your respective areas. So I guess, Martha, I'll start with you. The NIH is always, of course, the go-to place when these new things happen. You know, the government always turns to the NIH, National Institutes of Health. You know, we have so many initiatives going on, and I know Lucie cannot participate with us today, but you know, you're right there in the thick of things. And can you tell us, from your perspective, you know, amazing things that were done very rapidly, right? How was the NIH able to really pivot so quickly to bring regenerative medicine resources to the field?
Well, I think the CARES Act actually provided some resources for you know, moving forward. As you know, the long hauler effects of COVID-19 affect our major blood systems, heart, lung, and blood systems, as well as other systems, neuro and kidney. But I think it was, you know, recognizing the need that our communities are being overwhelmed with this virus, and also that it looks like there's some long-lasting lingering effects for those that were infected and were not protected from immunization, vaccination. So I think, and then there's just a general overall sense that this is what we need to do as part of our vision, our strategic vision. I think any feedback from this webinar will be helpful in, you know, redefining our strategic vision going forward for regarding COVID, as well as any possible future pandemics that could be on the horizon.
Great, thank you, Martha. Sohail, from CIRM, can you give me the same thing in terms of, I mean, from your perspective, in terms of what you have all been doing, and how you handle this in terms of COVID-19, also certainly rapid pivoting at your end.
Absolutely. Yeah, thank you, Anthony. So basically what's happening is that we have programs, as you know, the stem cell programs. And but our board, because we are a state agency and we are responsible for the health and safety of the people in California, so rapidly, I think our board approved that we need to utilize our resources and see what the investigators in California can do for the COVID-19. So in March, within 1 month, we were able to provide our resources and have our funding mechanism start. And generally, it used to take longer time for our funding mechanisms to do. But here, because of the urgency, within a month, we were able to have the board meeting, have our grants working group, and start funding research in both from discovery, translation, as well as a clinical trial. So in a short period of time, as I described in my presentation, we were able to launch 20 programs, and every month, 1 program each. And actually, we have been very fortunate because the investigators in California were very receptive to work on these programs. Some of them are stem cell-based programs. Some of them indeed are using small molecules, and also have programs which are using all different aspects, both from the discovery side and all the way to the clinical trials. And I think one thing which we really learned very quickly is that science is really needs more of a systems biology approach. We can't really work in isolation. There is a need for collaboration between virologists, stem cell scientists, and our people who are working on tissue engineering, and all of them can work together to make something happen which can really benefit. I mean, one example I think you are probably aware of is Derek Rossi. Derek Rossi was a stem cell scientist in our Weizmann's lab, and he was working, and then he thought that he can use messenger RNA to do reprogramming rather than using the DNA what Yamanaka was using. At that time, he never thought that the messenger RNA, which he's going to use for reprogramming stem cell iPSCs, would one day be used for making a vaccine. And now the Moderna vaccine is Derek Rossi's work, which he started out as a stem cell scientist in our Weizmann's lab. So I think science has ways to work out. And I think we are glad that we are able to help and enable some of the research here in California, which can be benefit not only to California, but all over the world. So I think we are very pleased that the investigators are really were able to do what they are really good at doing.
Thank you, Sohail. Now, Rick, you were right in the hotbed of activity when COVID first came into the U.S. I mean, the hospitals were packed. It was pandemonium, really, in New York City. Absolutely. And you were right there amidst all these things going on. So tell me, how did the New York Stem Cell Foundation actually start to rapidly deploy as it did also, and yeah, lessons learned?
Yeah, so I mean, of course, in the early days, we had to really ramp down our activities. There were so many restrictions on the number of people, but we were able to keep a skeleton crew on the ground. We did get a designation as an essential business due to some of our work with the National Institutes of Health. And so we were able to have some staff come on and keep things going. And as time went on, we reached out to our broad network across the country and, started talking to people about, what would be helpful for them in their research. And it became clear that our biobank was, sitting there with a lot of IPS lines that we had genetically characterized and could provide really great starting material for people to start looking at, infectivity and responses in real human cells and human cell derived models. And so we started to, explore that, and eventually we were able to get enough people on site through putting in, a variety of safety measures that we could start looking at the differentiations into lung type cells and ways we could leverage, the genomics we have in our biobank to study that further.
That's great. Thank you, Rick. And Mahendra, you've actually been in academia. You were at the NIH, then at the major foundation, and now in industry. So you've actually covered all areas in your career, and here you are now in industry, and you have basically this broad view, right, of all these things and how these things work. So from your perspective, do you think industry really stepped up to where it needed to go?
So the answer is yes, but before I talk about industry, I really wanted to put a plug in for the journals, and especially stem cell translational medicine as well. They did two really important things. One, they did this fast review for COVID-related articles, and they made them freely available for download. I can't tell you how important that was for being able to move research forward rapidly. And then there were efforts like what you saw, right, in industry, and your effort as well to put an industry coalition together to be able to provide resources and reagents. Because as Sohail said, it takes a village. And we were able to get reagents for ACE receptor co-expression, ACE2 receptor expression. We were able to make these pseudo lines for testing. We were able to use the vaccine-related proteins for testing from industry. So industry really stepped up, and even though they own certain things, they were willing to share. And that's really moved science forward incredibly rapidly. The NIH and BARDA really provided a lot of funding at a very rapid pace. I mean, we, for example, could not have done some of the work without the fact that we were able to access a small amount of money, not the big amount that we'd have liked, but nevertheless, it was really very nice to be able to do that. So I really think industry did step up, and it's actually amazing how much was done on that basis.
Yeah, I fully agree. I mean, here's a vaccine that got developed within 10 months, right? Unheard of, absolutely unheard of in the history of medicine, to turn around such quickly with new technologies, right? It couldn't have been possible without the RNA technology. But Mahendra, and thanks for the plug for stem cell translational medicine. I do think the journal did amazing things. A big thanks to our publisher, Ann Murphy, who and AlphaMed Press, who really pushed, led the way in that area as well. So basically, thanks for your answer in industry. I really do think they stepped up to the plate. So I guess the question really becomes, we have limited time. So what I'd like to really discuss now for the whole panel is lessons learned. Here we are, we were able to deploy a lot of regenerative medicine resources, a lot of reagents and know-how and knowledge base and funding all coming together to advance to the point where we are today in COVID. I guess at this point, I'd like to, and I'll start with Martha on this again. Okay, Martha, just because you're the brain power here, but basically, I would just say briefly, very briefly, lessons learned. What can we do for the next pandemic? Things that we
yeah, that's probably coming up sooner than we think. It's not going to be 100 years from now, probably. But yeah, I think lessons learned are better coordination, and at the federal level, I mean, it all depends on the top-down kind of leadership. I think Operation Warp Speed was a fantastic influx of support from the federal government, but we needed to have more of a overarching strategic plan for getting the vaccines out and doing additional research that's necessary to understand some of the long hauler effects. Just looking at children, I mean, children are dying also, and they have this multi-inflammatory syndrome thing that is kind of similar to Kawasaki disease with long-term effects on the heart, lung, and blood systems. So having a better sense of coordination, which I think, your efforts are really could be instrumental going forward in terms of having an overall one-stop shop for COVID resources. So I think, in collaboration, we could probably do better going forward, but I'm actually pretty impressed with the overall response NIH has been able to provide to address this pandemic. So that's kind of where I'm at.
Great, thank you, Martha. Appreciate that. And Sohail, how about from CIRM? Lessons learned, things for the next pandemic that we can rely on.
No, I think systems biology, I think it really come down to science and having people collaborate with each other. That is the lessons learned, and it has made so much difference. We never had people, neurologists working with the stem cell scientists identifying that ApoE4, which can increase chances of Alzheimer's disease. Now it turns out ApoE4 can also lead to the COVID-19 brain fog. And so there are a lot of lessons learned in terms of collaborations between scientists, between administrators, between funding agencies, between federal agencies, collaborations, system biology approach to identify and automation in technologies, messenger RNA, making a vaccine used to take 3 to 4 months to do it. Now you can do it in automation within 60 minutes. This is amazing. I think automation and technologies are needed for the next generation. So I think from, again, system biology approach, people working together and automation would change the way people are doing science.
Great, thank you, Sohail. Thank you so much, Rick. Same at your end from where
Yeah. I agree with Martha's point about the coordination, also amongst the basic scientists. So I think, we kind of know the questions that came up that had to be answered now, and having some pre-planning in place to deploy capabilities really rapidly to answer those questions, and also provide, good messaging to the public. So, early on there was so much conflicting messaging about the transmission paths and, whether to mask or not. And, knowing right away that those sort of things should be studied in a really serious way would be super helpful. And I think also then, at the more basic level, just having pre-established networks. I mean, it was amazing how quickly scientists came together and, opened up about what they were doing and shared reagents and materials. But, everyone kind of had their own idea here and there, and some overarching coordination to get to the big questions quickly, I think would be super helpful.
Terrific, thank you. And Mahendra, how about from your perspective?
I have two things, that really struck me. One was we learned that the way that we usually do things is not going to work when we have a crisis, and it exposed all our failures. And that was very important. But the positive thing for that was it also showed us what we need to do so that we will not have the same problems again. And I think this was true for both the government, as you already heard from Martha, but it was also true for the regulatory side of things. And I want to remind people of what the FDA said just yesterday. They said that when we have to make a vaccine against the variant strains, we don't have to go through the standard regulatory process. We should be able to approve it in a month or less than a month so that we could have booster shots available. They had to think out of the box, right, to be able to do that. And that's happened to a lot of industry and a lot of ways on how to apply technology. So I think that's the really important positive thing that has come out of it, is we have discovered new ways to do things which are cheaper, better, faster, but not necessarily any more risky or less safe, right? And we were forced to do it and think out of the box. And that, I think, was a real plus from what we learned.
Terrific. Thank you so much. I know that we're already a few minutes over because we were running a little bit late from the conference. So I don't want to keep everyone, too much longer. But this has been just a fabulous session. I really would like to thank all the panelists. I'd like to thank the Regenerative Medicine Manufacturing Society and Josh Huntsberger for all the efforts that he has led in to make this session possible. Same thing with Ann Murphy and the Journal of Stem Cells Translational Medicine, our publisher, AlphaMed Press. We are thankful to you for setting this up, and for your staff, for all your staff members as well, for both the RMMS and SCTM. And of course, to thank the participants today who have made this session possible. And finally, to thank our panelists. Thank you, Martha Lundberg, Sohail Talib, Mahendra Rao, and Rig Monsma. Thank you all so much. Your presentations and your discussion was just they were just fabulous. Thank you.
Thanks, Tony, for keeping us all honest.
Yep.
Yeah, exactly.
Thank you so much. So I guess I'll turn it back over then to our host, Josh Hansberger and Ann Murphy, for them to close out.
Great. Just a final thank you to all of our speakers. I think we had a wonderful, wonderful program. We will be following up with a perspective article that will cover all of the topics that were discussed today. So you can look forward to that coming out in Stem Cell Translational Medicine. And I also urge you to visit the Stem Cell Portal page and please post your resources, because this can be at least one way. One of the things that I learned is the importance of coordination and coordination of these resources, and this can be one way to help coordinate those resources and help with facilitating collaborations. So with that, everyone take care, be safe, and thank you again for your time.
Thanks.
And any
Thanks.
final,
Thank you.
and final words?
Bye.
Okay.
And do you have any final words?
I just wanted to say thank you for a wonderful panel and wonderful presentations. And I just love the lessons learned and what we can do in the next pandemic. You know, a great way to end. So thank you, everybody.
Thank you. Thanks, everyone.
Thanks. Bye.
Bye.